So very good evening everybody. Yes, so today I'm going to start with the next part of the biotechnology, and that is applications. That is about RNA interference. So what we have done in the very last class, we have talked about Bt cotton, if you remember. But before I start, I must give you some of the informations about Bt cotton first, and then I'm going to get into RNA interference, so that you understand the difference between Bt cotton and RNA interference. In RNA interference, you can see this. What is written over there? Pest resistant. Pest resistance. So we have already done this pest resistance in Bt cotton. If you remember, in Bt cotton, what we have done in Bt cotton, what is Bt stands for? It's a transgenic plant. Bt cotton plant is a transgenic plant. What is the meaning of transgenic plant? Where a plant wherein we have introduced another DNA of other organisms. So what is the organism in case of Bt cotton? In case of Bt cotton, what is the organism? The organism is Bacillus thuringiensis. The organism is Bacillus thuringiensis. Okay? It's Bacillus thuringiensis. Now, I just need to give you a basic idea about this Bt cotton first, and then move on into pest resistant variety, and that is we are going to talk about RNA interference. Fine. Chalo. The first one is about Bt. What is Bt stands for? Bt stands for a bacteria named as Bacillus thuringiensis. I'm going to wrap it up. Bacillus thuringiensis. Now this bacteria, the DNA of this bacteria has been introduced inside the cotton plant. So we have made this cotton plant a genetically modified plant. That means we have made this plant as a transgenic plant. Clear? GMO or GM plant. O for organism, genetically modified organism. So this is a genetically modified plant. So GM plant, or we can also call this plant as, very good, transgenic plant. So you can all use this term in your when you write about this particular thing, you can use it as a transgenic plant. Now, this is a Bt called Bacillus thuringiensis, which can normally, by the help of its cry gene, it can make, by transcription, it can make what? The mRNA, and this mRNA can make a protein, and this protein is called as crystalline protein. This protein is called as crystalline protein. So how to mention it as CRY, cry protein. And this crystalline protein is an insecticidal protein. What is it? It is an insecticidal protein. That means it is having insecticidal properties. That means it can kill the insects. It can kill the insects. Very good. Now, the process by which crygene can make mRNA is called transcription. It's called transcription. The process by which mRNA can make protein is called translation. Now, this Bt gene is introduced where? This Bt gene is introduced inside the plant, cotton plant. So how this introduction can be done? The introduction can be done by the help of two types of vector. One is agrobacterium. In case of plant cell, the vector can be Agrobacterium tumefaciens, or the vector can be a retrovirus, We can remove the RNA and introduce our gene of interest and allow the virus to infect the plant cell. So what it's going to do? It's going to put the gene of interest inside the plant cell. So, okay, tumefaciens. Now, this Bt cotton plant is already having the DNA of Bt, that is Bacillus thuringiensis. And ultimately, this protein, cry protein, which normally remains in inactivated condition because it's a prototoxin. It's a prototoxin. Proto means what? Prototoxin. What is the meaning of prototoxin? It's an inactive toxin. It only gets activated in presence of alkaline pH. Very good. In presence of alkaline pH, it only gets activated into active protein. And this active protein receptors are present in the pests, in the normal pest of cotton. The normal pest which is being defined in your book is two of them. One is called cotton bollworm. And the one is corn borer. Fine. So these are the insects which can come under the phylum Arthropoda, which can belong to phylum Arthropoda under different under different class, under different phylum, under different class, the class can be class insecta, and under class insecta, there are three particular orders that has been defined in your book. What are those? The first one is order, three orders. One is order Diptera, order Lepidoptera, and the third order is about order Coleoptera. Can you just help me with the examples of them? Tell me about Coleoptera an example. It's beetle. It's beetle. Tell me the examples of Lepidoptera. Under Lepidoptera. Under Coleoptera, there is another example last day we talked about. It is rice weevil. Rice weevil. Under Lepidoptera, the examples can be armyworm. It can be tobacco birdworm. And tell me the last. And cotton bollworm will be also under this. It's going to fall under this category. Cotton bollworm. Okay. About Diptera, di means what? Two. So they have got two pairs of wings. So the examples of them can be fly, they can be mosquito, they can be other insects like cockroach can be also under this. So cockroach can be also under this. So what is common with all these three orders under class insecta, under phylum Arthropoda? What is common? In the midgut, they have got three guts, three parts of the gut, foregut, midgut and hindgut. In the midgut, they have got called mesenteron. They have got a specific type of membrane which is called as peritrophic membrane, and in the midgut, as because they have got peritrophic membrane, mind you, the peritrophic membrane are having the receptors of this active toxin, receptors of cry protein. Very good. Active toxin which is cry protein. Now, whenever the particular insect is going to feed on any part of the cotton plant, so this is the foregut, this is midgut, and this is the hindgut. Now, in the midgut, they have got receptors, and this is peritrophic membrane where the receptors are present. Now, these are the salivary glands. The moment they're going to feed on the leaves or any parts of the cotton plant, as because this cotton plant is already having Bt gene called cry gene, so that remains in inactive form. But the moment they are going to feed on it, this particular salivary gland is going to secrete alkaline secretions, which is going to make it active toxin. And these active toxin receptors are there always where? On the peritrophic membrane. The moment the insecticidal protein is going to come over here, it's going to make instant pore inside this midgut. And it's going to make the pore inside this gut, and instantaneously it's going to kill the particular insect. Clear? This is the way that we have been specific towards the pest which we want to get rid of. We can kill those pests. This is called specific pest we are acting upon. This is done by a biotechnological product called BT cotton. So there are many of the BT. There are many plants having BT gene, like there are BT brinjal also. Fine. It is only going to harm these insects called the mainly pest of the cotton plant, called cotton bollworm or corn borer. What you have to remember is the gene which is there in your book called Cry1 AB, Cry2 AC and Cry2 AB. Cry1 AC, Cry2 AB and Cry1 AB. Fine. These two for cotton bollworm and this is for corn borer, I think. You have to remember this. These two genes can make that inactive toxin which can only get activated in presence of alkaline secretions. That's what we have talked about BT cotton. Today we are going to talk about RNA interference. A very, very important topic. It's important not only for your board exams. You can expect in your board exam about this question, RNA interference, and for NEET it's a very, very important one. Fine. So let us talk about RNA interference. What is the meaning of the term interference? What is the meaning of the term RNA interference? Come on, let's start with RNA interference. I think this is a very good term right now. You are using it with your parents, I think. Don't interfere. This is the right thing. This term is very good term for you. Okay? Interference. Now, interference means interfering with what? With RNA. Which RNA? mRNA. Which RNA? Okay. RNA interference. The other term for RNA interference is silencing of mRNA. What is mRNA? The messenger. mRNA is the messenger RNA. So normally we know if this is the DNA, if this is the DNA, 5 prime, 3 prime, 5 prime, 3 prime. Okay, this is the DNA, A, T, G, C, G, C, A, T. Okay, so this is the DNA strand, and it's going to make what? mRNA by the process of transcription. Tell me what is the outcome of it. Tell me the strands. Tell me the bases. This will be dealt. So it will be A, U, G, C. A, U, G, C. Is the mRNA strands? And this mRNA needs to reach where? Inside ribosome. Inside ribosome. Inside ribosome. Inside ribosome. Ribosome is having how many subunits? 2 subunits. Ribosome is having how many subunits? 2 subunits. This is the ribosome having 2 subunits. Fine. This mRNA is going to go over here. It will reach over here, 5 prime, 3 prime. This is the larger subunit, small subunit. This is having P site, A site, or we can also add another site called E site, EPA. Not required. P and A. Okay. Now it is going to make what? Protein. It's going to make what? Protein. So this mRNA is going to reach in the ribosome and make the protein. This is what process called? This process is called translation, and this is called transcription. This is called transcription. This is the basic idea about central dogma that we know, and DNA also replicates itself by the process of DNA replication. These 3 together is called central dogma. It was given by Francis Crick. It was given by Francis Crick. Okay. Now, the purpose of RNA interference and making pest-resistant plant. See what is written over there? Pest-resistant plant. Let me first give you a basic idea. Then we are going to go in depth of it. Fine. NCERT के लाइन बाय लाइन में जो इन्फॉर्मेशन है वो पहले जाएंगे, उसके बाद हम इन्फॉर्मेशन थोड़ा डेप्थ में जाएंगे. Okay, which is going to help you to understand it completely. Pest resistant plant. Find out the term in your book where it is written novel strategy. Find out and underline the term novel strategy. So in your board exam, what is the question going to be? You're not going to expect straight away questions. Explain RNA interference. No. They want to ask you, explain a novel strategy to protect a plant from pest. That means you have to talk about RNA interference. The second term, find out in your book, called cellular defense. Cellular defense, and underline it. Cellular defense under pest-resistant plants. Cellular defense. It's a very, very important term again. Cellular defense. Very, very important. Is this clear? Now, so in these 2 terms are very, very important. Why it is a novel strategy? Why it is a novel strategy? In BT cotton case, you have killed the pest or you have helped the pest understand you should not do this. This is very wrong from your part. Your parents never taught you not to feed on this kind of plant and affect the cotton yield. Aisa tha? No, you have killed them. But here you have taken a novel strategy. Kya novel tha? What is novel in this strategy? You never wanted to kill that particular worm. Fine? घर में पेट है, पेट कुत्ता है, पिल्ला जो है पिल्ला, तो वो पिल्ला जब आपको बाइट करता है तो वो सोचता है कि क्या बाइट कर रहे हैं, बिकॉज वो अपना स्ट्रेंथ दिखाएगा. वो तो खेल रहा है ना, उसका तो और तो दोस्त नहीं दिया आपने. एक ही पिल्ले को आपने घर में रख दिया तो वो तो क्या करेगा, पहले तो आपका चमड़े का जो चप्पल है उसको फाड़ेगा वो, टू शो ऑफ, क्योंकि उसमें एनर्जी आता है तो है ना. And when it tries to bite you, you keep your fingers in such a way that the canine gets within the fingers. and they give a bite. तो आप जानते हो ना, तो बाइट तो नहीं होगा बिकॉज वो कैनाइन जा रहा कहाँ पे है और वो सोच रहा है, देख रहा है, अरे चिल्ला कुछ, इतना जोर से बाइट किया. सो यू आर नॉट बीटिंग द पिल्ला, यू आर हेल्पिंग द पिल्ला टू गेट सम एनर्जी. एंड जब अगर रियल बाइट हो तो समझो कैसे बाइट करना है. तो आपने क्या नहीं किया, पिल्ला को मारा नहीं. यहाँ पे व्हाट इज नॉवेल स्ट्रेटेजी? यू आर नॉट किलिंग द पेस्ट, यू आर सेविंग द प्लांट फ्रॉम द पेस्ट इन्फेस्टेशन बट नॉट किलिंग द पेस्ट. बाबा समझ में आ गया कि नहीं आया? ये नॉवेल हुआ नहीं हुआ? বুঝেছিস না বুঝিস নি? ও তোকে এফেক্ট করবে, তুই বলবি খুব ভালো লাগছে না এফেক্ট করে, ভেরি গুড. ইউ আর সেভিং ইউর সেলফ বাট নট by not killing the particular one which is affecting you. It's a novel strategy and it's also a cellular defense. How? First understand, see, in this whole concept, RNA interference means you are interfering with the RNA. You are interfering with the RNA. You are making this mRNA double-stranded. You are making this mRNA double-stranded. If the mRNA becomes double-stranded, do you think it is going to be able to reach to the ribosome and make the protein? This is how you are silencing the mRNA. Very basic, huh? We are going to go in depth of it. So what is the meaning of mRNA silencing? mRNA silencing or RNA interference. You are interfering RNA so that it is never being able to reach to the kitchen to make the protein. What is the kitchen? Ribosome. Is this clear? Tell me the actor, the famous statement of the actor was, famous dialogue was, Khamosh. Shatrughan Sinha. So you are khamoshing whom? mRNA. Not allowing the mRNA to reach where? Inside ribosome. In a broad spectrum, this is the answer. But we are going to get in details of it. Edu clear ache? Kono oshubidha ache baba? Any issues with this? So khamoshing of mRNA. By making it double-stranded. By making it double-stranded. Let's get into the details of it. Translation hobe na. Translation ta hote debe na. Translation hole ki তৈরি করবে? প্রোটিন তৈরি করবে. Now understand what is that protein? What is the best if you get bitten by a snake? What is that snake venom contains? It's a proteinaceous. It is so very good protein that you cannot digest it in your living state. So you have to go up there. You have to take some time to digest it. So it's a protein. So think about it. In this RNA interference, we have to talk about a plant called tobacco plant. So let's get into the details of it. So what is the name of the plant? Tobacco plant. Now tobacco is very useful, not for you. Tobacco is very useful. It's a farmer when grows the tobacco plant, if it is being infested by a type of worm. So what is the normal pest of this tobacco plant? The normal pest of this tobacco plant is a worm. Pest of this tobacco plant is a worm, and the worm is a nematode. Worm is a nematode. What is nematode? Tell me what is nematode. If you remember animal kingdom mone ache? Tell me which comes under which phylum? Ash helminthes. Nematoda. Ash. What is the phylum? The phylum is Ash helminthes. Ash helminthes. The phylum is Ash helminthes. Ash means round and helminthes means worm. So it's a round worm. Tell me some of the examples of round worm. The basic example was Ascaris lumbricoides. Very good. Ascaris lumbricoides. They have got sexual dimorphism. They have two different body, two different sex. The male one was smaller and having pointed ends, and in the point they have got one particular structure called pineal city, which acted as a copulatory organ, and in the female body is longer and elongated. Kire mone asche? Sexual dimorphism we have studied. Hai na? Now Ascaris lumbricoides. It creates a disease called ascariasis. It makes a disease called ascariasis. So ये सब एनिमल में होता है सर, कहाँ से प्लांट में आ गया? We are going to talk about. इतना नॉर्मल नहीं है. But for tobacco plant, the infestation by nematode is a very normal one. ये बहुत नॉर्मल है for tobacco plant. तो ये तो एनिमल में, ह्यूमन में इन्फेक्ट किया ascaris ने. Okay, now ascaris हो गया. Tell me some more examples of it. Wuchereria bancrofti. Wuchereria bancrofti. Tell me. Elephantiasis. Elephantiasis. It creates elephantiasis. Filariasis also. Elephantiasis. We can call it as filariasis also. Fine. Tell me the vector for this.
Mosquito.
Which mosquito? Aedes aegypti. Culex? Wait. Aedes is dengue. Culex, rightly said. Culex fatigans. Culex fatigans is the vector. Aedes causes dengue. It acts as a vector. Any other example? Any other example of Loa? Eyeworm, remember? Loa, eyeworm. Noah. Good. Now you remember it. Eyeworm. And any other example? Ankylostoma duodenale, hookworm. Ankylostoma duodenale, it's a hookworm. Oh sir, we are going away from RNA interference. Let's come back. Okay, so this is hookworm. Now, see, the name of this nematode or the name of this particular nematode which infests this tobacco plant is Melodigine incognitia. This is the name of the nematode which infect mainly the tobacco plants. Which part? Shoot or root? Or flowers? Root. It infects the roots of the tobacco plant. If the tobacco plant root gets infested, if this is the root and this root is being infested by, this is the nematode. This is the nematode. It is not looking like snake, no? It's nematode. It's a nematode called Melodigine incognitia. The moment this particular worm is going to infest this particular cells, do you think that tobacco, what is the best part of the tobacco plant when you're yielding it? Leaves. Do you think the leaves are, this plant is going to have huge number of leaves if the root is being infested? No. So you have to stop the infestation of this nematode, but not by killing nematode. That is a novel strategy. Nematode খাবে একে, কিন্তু এর কোনো ইনফেক্ট হবে না, এর কোনো ক্ষতি হবে না. Yes. Clear? বাবা বুঝলাম না? And how we can do it? See. Silencing the mRNA of whom? Tobacco plant or nematode? Nematode. Because nematode is going to make the protein. Nematode in its body is going to make the protein which is going to actually harm the cells of tobacco cells or tobacco roots. So we have to stop forming what? We have to stop forming protein of whom? Of the nematode. Because this protein is going to act as a toxin. And this protein will be formed by, Alisha help me. This protein will be formed by mRNA. And this mRNA will be formed by the specific part of the DNA of the nematode. The specific part of the DNA of the nematode. Clear? This is DNA. And this DNA by transcription is going to make mRNA. And this mRNA by translation should be making what? This toxin. By any means, if we can khamosh this mRNA by making a double-stranded what is it going to be? This is going to be UTACG. If this is the mRNA, then we are going to make a double-stranded RNA if we can make it. And we can stop the translation process. Ultimately, toxin produce nahi hoga. Toxin is not going to be able to be produced inside the nematode. Clear? So nematode is not going to be able to infect the particular host cell, this particular tobacco plant. Any issues with this? Clear? So do you think in any case we are harming this nematode? No. Any issues with this? Okay. Okay. So how we are going to do it? First, we have to make this particular plant. What is this plant? Tobacco plant. What is this plant? Tobacco plant. Okay. So we have to make this tobacco plant a transgenic one. Tobacco plant को क्या बनाना है? We have to make this tobacco plant as a transgenic. किसका जीन इसके अंदर इंट्रोड्यूस करना है? Find out in your NCERT the whole genome of nematode. The whole genome of nematode which can make the protein, which can make the toxin protein. We have to introduce the whole genome of this nematode inside the tobacco plant. Inside the tobacco plant. Now, yes. How you are going to introduce it? क्वेश्चन समझ में आ गया? आपको क्या करना है? सोचो, ये आपका एक बुक है. ये आपका एक बुक है. हाँ? बुक है. बुक में 10 पेजेस है. 10 पेज में आप बहुत सारे हॉरर मूवी में देख लिया आपने ये. हाँ, बहुत सारे फिगर रहेंगे. ये फिगर देखने के बाद आपको आएगा. हाँ, ये बुक खोलने से हो जाता है. ऑल दिस हिंदी मूवीज, बॉलीवुड मूवीज. नाउ थिंक अबाउट इट. द होल बुक के अंदर कौन सा पेज इज गोइंग टू अल्टीमेटली हेल्प इन द फॉर्मेशन ऑफ टॉक्सिन? यू डोंट नो. You don't know that. So what you have to do is you have to introduce the whole book inside the tobacco plant. उसमें से एक पेज, हर एक पेज तो ट्रांसक्रिप्शन और ट्रांसलेशन होगा. आउट ऑफ दैट वो पेज जो टॉक्सिन बनाता है वो भी तो हो जाएगा बाबा. Any issues? Find out where it is written. Whole genome of nematode specific gene is being introduced. Nematode specific gene in the host plant. Nematode specific gene is being introduced into the host plant. Have you got it? Yes. Clear? Now, Karachi. Good. Now, congratulations. Tobacco plant transgenic. So you have made the tobacco plant transgenic by introducing this whole genome. By introducing this whole genome, where whole genome inside the bacteria plasmid. What is the vector? The vector is agrobacterium. The vector is Agrobacterium tumefaciens. Agrobacterium tumefaciens. Rajendra is going to tell me what is the name of this plasmid, of this bacteria. This is the bacteria. इसका plasmid का नाम क्या है? What is the name of this plasmid? The name of the plasmid is TI plasmid. What is TI stands for? Tumor inducing plasmid. TI plasmid is for tumor inducing plasmid. What is the natural? This particular bacteria is used as a natural vector for all plants. We have got two ways. Either we can take the Agrobacterium tumefaciens as our main vector, or we can also take the use of retrovirus to introduce our gene of interest inside the plant cells. Inside the plant cells. Is this clear? Now, inside the TI plasmid, what you have done? You have introduced your entire genome of the nematode specific gene inside the tobacco plant by helping this Agrobacterium tumefaciens infect the tobacco plant. The moment this bacteria is going to infect the tobacco plant, it's going to introduce the entire genome of the nematode. Is this clear? So now the tobacco plant is having the nematode specific DNA or not? Yes. And a nematode specific gene or not? Yes, it is having it. So what kind of tobacco plant we are going to call it as? Normal tobacco plant or a transgenic one? किसका जीन है उसके अंदर? अरे हाँ, एग्रोबैक्टीरियम का जीन है. this gene is of nematode specific DNA. So nematode का जीन अभी tobacco plant के अंदर है. Any issues? पहले से आपने रख दिया. हर एक सेल में this particular book is there. और बुक का कौन सा पेज टॉक्सिन करता है? कोई एक पेज. Any issues with this? Very good. Now this concept of RNA interference, this concept of RNA interference was discovered by two scientists called Fire and Mello. In the year 2006. And they got Nobel Prize. In the year 2006. In the year 2006. Fire and Mello, they got Nobel Prize. Okay. They got Nobel Prize. Now, RNAi, what is the concept of it? What is the concept of it? Understand. Now we are going to get into the details of it. Let us get into the details. Now see. See. Yes. This is the particular DNA. This is the DNA. 5 prime, 3 prime, 5 prime, 3 prime. Normally it makes what? Normally it makes single strand mRNA. Normally it makes what? Single strand mRNA. If this plant is of transgenic, if the plant is transgenic plant, in the transgenic plant, when this particular DNA is going to transcribe, called the process called transcription, it's going to make double stranded RNA, dsRNA. It's going to make what? Double stranded RNA, and this is going to be complementary. This is going to be complementary. Come on, make it complementary. AUGC. Come on, GCAU. This is complementary. Mind you, out of this double stranded RNA, one of the strand is sense strand and another strand is antisense strand. And the strand is, don't make it nonsense, it is antisense. One is sense and the one is antisense strand. So how many strands are there in this RNA? Double strand RNA. Fine. Now, this particular double stranded RNA will be, so double strand, triple strand. This long double stranded RNA will be cut by, let us make it like this vertically. This is 5 prime, 3 prime, this is 5 prime, 3 prime. And there's a bond in between, and this is dsRNA. This will be cut into pieces. This will be cut into pieces by a protein called dicer protein. By a protein called dicer protein into small segments. Into small segments. 1, 2, 3, 4. So there are 4 pieces. Say for example. Clear? I'm not putting the conventions 5 prime, 3 prime, 5 prime, 3 prime. It's very obvious. 5 prime, 3 prime. Clear? 5 prime, 3 prime, 5 prime, 3 prime. This is quite obvious. Okay. So these are what? The small pieces. The small pieces of this double strand RNA are called as siRNA. What is siRNA? What is siRNA? What is the meaning of this? Single. What is this I called? Si called? No. No. Small interfering RNA. Small interfering RNA. So these small pieces of interfering RNA are present where? In the host cell. What is the host? Tobacco plant? What is the host cell? Tobacco plant? So tobacco plant में ये है. Clear? Tobacco plant में ये सब small. Now they are having this siRNA, small piece of RNA, small interfering RNA. What happened? Tobacco ज़्यादा हो गया. अच्छा कोको हो गया. So tobacco देखा एक लूज बॉल मिलेगा तो छक्का मारना ही है. अच्छा tobacco plant. So this is tobacco plant, and inside the tobacco plant what has happened? By the help of dicer protein, you have got certain small pieces of siRNA. These are small interfering RNA. Now this particular plant cell, ये plant cell के अंदर है, ये plant cell के अंदर हो रहा है, and this plant cell is now infested by, ये आ गया हमारा villain, कौन है ये? This is nematode. This is nematode. A nematode जब plant cell को खाएगा, so what is going to get inside? This small piece is going to get inside. Come on. This small piece is going to get inside. That means siRNA को उसने खा लिया. स्मॉल इंटरफेरिंग आरएनए को उसने खा लिया। यहां तक क्लियर है? जस्ट हैव अ लुक, यहां तक क्लियर है? यस। एंड देन व्हाट इज गोइंग टू हैपन? व्हाट इज गोइंग टू हैपन? सो किसने काटा इसको? डाइसर ने। किसने काटा इसको? डाइसर ने। नाउ निमाटोड इज हैविंग siRNA निमाटोड के बॉडी में चला गया, तो दिस इज आवर विलेन कॉल्ड nematode, and inside nematode they have got what? siRNA. Now this is what is siRNA stand for? Again, not small interfering RNA. अच्छा, ये तो इंटरफर करेगा कहीं, कहां पे? इसका मेन काम क्या है? व्हाट इज द फंक्शन ऑफ इट? इनसाइड दिस नेमाटोड हर एक सेल में क्या है? बुक है या पेज है? बुक है। हर एक सेल में क्या है? बुक है। व्हाट इज द बुक? द एंटायर जीनोम इज प्रेजेंट इनसाइड द नेमाटोड सेल्स। सो अब क्या करेगा दिस siRNA? ये जब this particular cell, normal cells of nematode, when they are going to start making their own, उनका DNA, जब उनका DNA, the moment they start making the mRNA, what is going to happen? ये उसका mRNA है, ये उसका mRNA 5' to 3', fine. इसी का the complementary basis of this mRNA is present in siRNA or not? है क्या? नहीं है। But this siRNA is a double stranded one, so double strand won't be able to make double strand, इसको खामोश नहीं कर पाएगा। Again I repeat, this is the entire genome of the nematode. This nematode is going to make mRNA, single stranded mRNA. But what you want to do? You want to make silencing of this mRNA. ये mRNA अगर, if this mRNA is able to reach up to the ribosome, then ये क्या बनाएगा? Ultimately protein. जो protein क्या बनेगा? Protein is toxin, and this is going to be acting as toxin for tobacco root cells. बोलो समझ में आया कि नहीं आया? तो कहां पे इंटरफेयर करना पड़ेगा? यहां पे इंटरफेयर करना पड़ेगा। पर अभी इसके बॉडी में क्या है? siRNA is present inside this nematode body. What is going to happen? See, this small siRNA, this is siRNA. देखो, this is siRNA. This siRNA is going to bind with another complex called RISC. क्या नाम है इसका? RISC. And what is the full form of RISC? RNA induced silencing complex. RISC. What is it? RNA induced silencing. RNA induced silencing complex. The moment it binds with this is going to, RISC is going to cleave one of the strand. इसका एक strand को cleave कर देगा। कौन सा strand को? Anti-sense strand को वो काट देगा, and sense strand को वो रख देगा। This sense strand is going to come and bind with, देखो this sense strand, इसको unit समझ में आया क्या? It's going to come and bind with the mRNA. This sense strand is going to bind with the mRNA and is going to bind with mRNA and make a double stranded. Again I repeat, RNA induced silencing complex is going to break these two strands. यहाँ पे क्या था? एक sense और एक antisense. ये क्या करेगा? sense को निकाल देगा। This antisense is going to come and bind with over here, because this is the mRNA, and it's going to bind over here and make it double stranded. क्या बनाएगा? और कहां पे खामोश कर दिया? कौन से जीन को खामोश कर दिया? एंटायर जीनोम को, या वो पेज को जो पेज टॉक्सिन बनाएगा? ये तो एंटायर बुक था। बोलो समझ में आ गया? एंड ऑल्सो द मोमेंट RISC रिमेन्स अटैच्ड विद इट, ये तो ब्लॉक तो करेगा, ब्लॉक करने के बाद ये दोनों को काट भी देगा। इट्स गोइंग टू कट ऑफ द होल mRNA को सिंगल पीस में तोड़ देगा, छोटे छोटे पीस में तोड़ देगा। इट्स गोइंग टू क्लीव द mRNA टू स्मॉल पीसेस and never allow this particular amine to make any proteins. और उसमें कौन सा प्रोटीन भी नहीं बना, द टॉक्सिन. सो क्या कर दिया उसने? खामोश कर दिया. What it has made? It has silenced the mRNA. It has silenced the mRNA. So what is the most important thing? Number one, just repeat it. First, we have made the tobacco plant genetically engineered by introducing the whole nematode genome inside the tobacco plant by the help of Agrobacterium tumefaciens. Now the plant is having the whole genome of the nematode sequences. And the whole genome means entire genome. But out of this entire genome, the portion of the DNA which can make the toxin, वो क्या बनाएगा? वो तो toxin बनाएगा. वो भी है उसके अंदर. So whenever a nematode comes and tries to feed on it, inside the transgenic plant, when that particular DNA starts making mRNA, it always makes double-stranded RNA, one sense strand, having a sense strand and having another antisense strand. But there is a protein called dicer protein. The dicer protein cleaves the particular double-strand RNA into small pieces, and the small pieces are called siRNA. क्या नाम है? siRNA. Small interfering RNA. Those remains inside the tobacco root cells. Whenever a nematode tries to feed on the plant cells, also takes up siRNA. The siRNA, when gets inside the nematode normally, by the help of the entire genome, is going to make a single-strand mRNA. But the siRNA, which is being taken up by the nematode, binds with RISC, that is RNA-induced silencing complex, and cleaves one of the two double-strand RNA into single strand, and it mainly cuts off the sense and antisense. The antisense is going to bind with this particular sense of mRNA and makes what? Double strand, because it's going to have complementary bases of this. अगर यहाँ पे A है तो इसके पास क्या है? U है. यहाँ पे G है तो यहाँ पे क्या? C है. And they're going to bind, and this portion, when it binds, not only it silences the mRNA, silence तो करेगा, खामोश तो करेगा, और करने के बाद वो छोटे छोटे पीस में, टुकड़े में तोड़ देगा इसको, the total mRNA. And ultimately what is going to happen? There will be no protein formation which is going to help the plant from this particular infestation by this nematode. Is this clear?
Sir, antisense का सेंस आलादा होएगा, antisense तो चलेगा, लेबर सेंस टा की कोड.
सेंस टा नॉर्मली खेद है, और कोनो काज नहीं. सिंगल स्टैंडर्ड कोनो काज नहीं ना.
सेंस का तो कोनो आइडिया
नहीं. आस्क दो ना. Clear? Is this clear? Let's find out over here in the what your book has to say about pest-resistant plants. Any issues with this? Come on. Any issues? बोलो समझ में आ गया नहीं है? Yes. देखो इधर. Pest-resistant plants. Several nematodes parasitize a wide variety of plants and animals, including human beings. आप जानते हो? Human beings. हाँ? Yes. Now, a nematode, Milordigini incognitia, infects the roots of tobacco plants and cause a great reduction in yield. 2016 question. Okay. A novel strategy was adopted to prevent this infestation, which was based on the process of RNAi in RNA interference in 2020. RNAi takes place in all eukaryotic organisms as a method of cellular defense. Again, cellular defense. So two terms are most important. One is cellular defense and the one is novel strategy. These two terms, whenever you are being asked with this, मतलब क्या मन में आना चाहिए? They're talking about RNA interference. This method involves the silencing of a specific mRNA due to the complementary double-stranded RNA molecule that binds to and prevents translation of the mRNA or silencing. See 2013 and 2022. The source of this complementary RNA, very, very important from here. The source of this complementary RNA could be from an infection by viruses. What is a virus? Retrovirus. Having RNA genome, that means retrovirus. RNA having, virus having RNA, it's called retrovirus. Or mobile genetic elements called transposons. Now what are transposons? Very, very important. What are transposons? Transposons are mobile genetic elements. That means this can create, if there has been mutation, it can help in inversion, it can help in deletion, Deletion. I think you know what is deletion. Tell me. Deletion of one of the base. We have seen this in sickle cell anemia. Point mutation. So this transposons are the pieces of the DNA. We call them as a jumping gene also. We call them as a jumping gene also. So we can always use this kind of transposons in biotechnology, where it's going to replace some of the fragments of the DNA. Either it is going to solve mutation or it's going to create mutation, whatever role it's being said to do. It's going to do that. But it's also called as jumping gene. It's also called as jumping gene. I can remember jumping gene was discovered by McClintock. It's a very, quite often question. This was asked in our times. McClintock, the person who discovered jumping gene. That replicate via an RNA intermediate. What is the meaning of RNA intermediate? That means this particular RNA genome can get replicated by an RNA intermediate. That means some of the RNA from outside is being introduced. Some of the RNA has been introduced from outside, which is called as RNA intermediate. Using agrobacterium vector, nematode-specific genes were introduced into the host plant. Come on, what is this called? Nematode-specific genes were introduced into the host plant. What is a host plant? Tobacco plant is the host plant over here. The introduction of DNA was such that it produced both sense and antisense RNA in the host cell. Both what? Sense and antisense RNA inside the host cell. So both of this double-stranded dsRNA went inside the host cell. These two RNAs, being complement to each other, formed a double-stranded dsRNA that initiated RNAi, thus silenced the specific mRNA of the nematode. But how? It's not being there in your book, so it's completely NCERT. So you cannot go out of the NCERT, but you have got the idea what is called RISC. You got what is the meaning of dicer. Clear? And what is siRNA? Okay. The consequence was that the parasite could not survive in a transgenic host expressing specific interfering RNA. The transgenic plant therefore got itself protected from the parasite. So the transgenic plant has got self-protected. Come on, self-protected from the parasite. ठीक है? तो ये पैरासाइट कौन है? ये भूत है. तो आपने घर में क्या किया? आपने भूत को आने के लिए कुछ मंत्र कर दिया, तो भूत आ नहीं पा रहा है. So आप क्या भूत को मार रहे हो? नहीं मार रहे हो. But you are protecting yourself. What is the भूत out here? Is a melodyne incognitia. Melodyne incognitia. Any issues with this? You got any question you can ask me? Can we solve some of the questions asked from RNA interference? Let us solve some of the questions. Okay. Yes. Okay. Let us solve some of the topics and then we are going to solve some of the questions. Biological applications in medicine. Babu, yahan tak clear hai na? Any issues with this? Let's find out what the NCERT has to say. The DNA technological process have made immenses. What is immenses? Immenses in the past in the area of healthcare by enabling mass production of safe and more effective therapeutic drugs. Further, the recombinant therapeutics do not induce unwanted immunological responses as it is common in case of similar products isolated from non-human resources. किसके बारे में बहुत बात किया जा रहा है? What is this they are talking about? Come on, non-human. What is non-human? Remember, insulin was earlier. Insulin 1920s. Insulin was for the human who were suffering from diabetes mellitus were administered with insulin taken out from the pancreas of dead cattle and pigs. Fine. So non-human था. उसके बाद आपने किससे बनाया ये? By the help of biotechnology, you have taken the help of bacteria and you use your gene of interest inside the bacteria, that is insulin forming DNA, and ultimately made the particular humulin or insulin. We will talk about that. At present, about 30 recombinant therapeutics have been approved for human use. The world over, in India, 12 of these are presently being marketed. One of them is humulin. So genetically engineered insulin. I'm going to do that first and then solve some questions, or solve kar le pehle? Solve some questions. Okay, no issues. Yes. Solve some questions. Ethical issues. Now we are going to get questions. Yes. Try with this. What is this? Given below are two statements. Yes. Up till this. Come on. ठीक से पढ़ो. Okay. Oh God. अच्छा क्या है? Statement पढ़ो. What is the statement? Completely NCERT based question. Transfer RNAs and ribosomal RNA do not interact with mRNA. Transfer RNAs and ribosomal RNAs do not interact with mRNA. Statement 2. RNA interference I takes place in all eukaryotic organisms as a method of cellular defenses. In the light of the above statements, choose the most appropriate answer from the options given below. Option? Statement 1 is incorrect, but statement 2 is correct. RNA interference takes place in all eukaryotic organisms as a method of cellular defense. Transfer RNAs and ribosomal RNA do not interact with mRNA. Do not or they do? They do. Why? Because transfer RNA, that is tRNA. Come on, tRNA, remember? Cloverleaf model. tRNA is what? Cloverleaf model. Come on. Cloverleaf model. This is what tRNA is. Fine. This is anticodon site. Come on. This is, what is this? DHU. DHU. This is what? T is IC. And this is amino acid binding site. Come on. This is 5' end. This is 3' end of the DNA. They have got loop. Why? Because they have unpaired bases. Come on. We have done it already in molecular basis of inheritance. So tRNA. tRNA किसको लाएगा? एंटीकोडॉन को लाएगा. एंटीकोडॉन किसके ऊपर बाइंड करेगा? एमआरएनए के ऊपर. और ये किसके ऊपर खड़ा है? राइबोसोम. और राइबोसोम किससे बना हुआ है? द स्ट्रक्चर ऑफ द राइबोसोम इज मेड अप ऑफ rRNA, राइबोसोमल RNA. तीनों को चाहिए बट दे डू नॉट. ये आंसर क्या है? ट्रांसफर RNA एंड राइबोसोमल RNA डू नॉट इंटरैक्ट विद mRNA. Wrong. Incorrect. So answer? Good. Good. Very good. Congratulations. Silencing of specific mRNA is possible via RNAi because silencing of specific mRNA is possible via RNAi because of silencing of specific mRNA is possible via RNAi because complementary dsRNA, inhibitory, complementary tRNA, non-complementary ssRNA. What is the answer? dsRNA. The BT toxin in genetically, ये बोलो, फिर से बोलो, creating pores in the midgut. What is the membrane? Peritrophic membrane. Following are the steps involved in action of toxin in BT cotton. वो आंसर था. आंसर क्या था? अरे क्या है? What is the question? Choose the correct sequence of steps from the options given below. Choose the correct sequence of steps. B-E-C. Okay, C, alkaline pH. I don't think so. The inactive, when they are talking about toxin, the inactive toxin converted into active form due to the alkaline pH. E के बाद A होगा. कहाँ पे E के बाद A है? E के बाद A कहाँ पे है? नहीं? हाँ? B.
और inactive toxin converted into active form तो पढ़े होच्छे.
Okay.
Conversion. ताले A B के start होगा. D.
B.
माने E option D होगा.
Option D. B first. Bacillus thuringiensis modifications with. Okay. Then E. The toxin proteins exist as inactive protoxin. ठीक है? C. The alkaline pH solubilizes the crystals. ठीक है? A. The inactive toxin. ठीक है? And last D. The activated toxin binds to the surface of beta gut. Good है. Very good. The capacity to generate a whole plant from any cell of the plant is called. Oh, we haven't done it, but answer is 20 potency. Transposons, bolo, can be used during which one of the following? Very good. Gene silencing. Transposon, gene silencing. In RNAi, the genes are silenced using double-strand RNA. Sir, ये तो बहुत गुल्ली डंडा हो गया सर हमारे लिए DSRNA. Which of the following is true for golden rice? It is vitamin A increased with the gene from daffodil. Good. What triggers activation of protoxin to active? Alkaline pH of the gut. What triggers activation of protoxin to active? अरे दो बार हो गया एक ही क्वेश्चन. Melodigium incognitia. बोलो. Which part of the tobacco plant? Melodigium incognitia. It is the root of tobacco. The crops engineered for glyphosate are resistant tolerant to herbicides. Kano? We have talked about enol pyruvate shikimate phosphate. These are glycophosphates. Remember, being a farmer, I have sprayed weedicides. Some of the weeds got resistant. Some of the main crops got resistant from that. So we have found out, some of the weeds got resistant. So we have found out which part of the gene can make it resistant. And we have found it, what was that product? Enol pyruvate shikimate phosphate. Enol pyruvate shikimate phosphate. These are nothing but glycophosphates. So answer is herbicides. Answer is herbicides. Bolo ye, ho gaya. Alkaline pH of the insect gut. Golden rice. Vitamin A. Which of the following BT crops is being grown in India? Cotton. Iron interference involves? ठीक से पढ़ो. Silencing of a specific mRNA due to its complementary consumption of which one of the following food can prevent the kind of blindness. What is the blindness called? Tell me the other term for that. The other term for that is xerophthalmia. Night blindness. Tobacco plants resistant to a nematode have been developed by the introduction of DNA that produced in the host cells. Bolo. Both sense and antisense. The process of RNA interference has been used in the development of plants resistant. Great. Silencing of mRNA has been used in producing transgenic plants. Nematodes. Tell me the name of the nematode. बोलो स्पेलिंग बोलो मैं लिख रहा हूँ. Melodigium incognitia. Chalo, very good. Okay. Now, humulin, genetically engineered insulin called humulin. What is the name of the company? Eli Lilly company. What is the name of the company? Eli Lilly. Find it out from your book. Find out. Eli Lilly company. क्या नाम है? Eli Lilly company. Eli Lilly company. Fine. Eli Lilly company. What is it? It's like this. It's like this. ऐसा है? ऐसा है ना diagram? This portion is A. Yes. A. नीचे B. अंदर C. ये B है. And this is C. And this is having disulfide bond. What is this called? SS bond. Remember that? Where we have studied a disulfide bond? Tertiary structure of the protein. Tertiary structure of the protein. We have studied about disulfide bond. Quaternary, tertiary. They have tertiary, especially tertiary. Like enzymes, all enzymes they have got disulfide bonds, disulfide linkages. Even in disease chapter we have studied about immunity. Under immunity, antibody. Remember? In antibody they have got, come on, this is antibody. Heavy chain and light chain. Heavy chain and light chain. So heavy chain they are having disulfide bond. Come on. So this is disulfide. What is this? This is called as proinsulin. This is called as Pro-insulin. Pro-insulin. Fine. Where from this concept has come? Come on. Let us start the concept from. Tell me the name of the organ which secretes insulin. Which cell pancreas से बनता है? Insulin pancreas से बनता है. Tell me which cells of the pancreas? There are some specialized cells. The cells are called islets of Langerhans. Langerhans was the name of the scientist. Islets of Langerhans. Islets means islands. So they were island-like cells. And these cells, they are having alpha, beta, gamma, delta. But we are going to talk about alpha, beta, and come on, delta. Okay, delta. बात नहीं करना है हमें करना है इसके बारे में बीटा सेल्स ऑफ islets of Langerhans. It secretes normally a hormone which is called insulin. And this insulin, what it can do is it can reduce the glucose level. It can reduce the glucose level. अब देखो ये है blood vessel. Now this is the blood vessel. This is the blood vessel, and these are the normal cells. These are the normal cells. Okay. These are the normal cells. अब देखो यहां से ब्लड वेसल से इंसुलिन जा रहा है एंड दिस इंसुलिन, अच्छा ये ब्लड वेसल ग्लूकोज है, क्या है ये ग्लूकोज है, हाँ इट्स रिच. ग्लूकोज कहां से आया? बाय ब्रेकिंग डाउन व्हाट? बाय ब्रेकिंग डाउन ग्लाइकोजन. बाय ब्रेकिंग डाउन ग्लाइकोजन. सो ग्लाइकोजन में क्या होगा? ग्लाइकोजन आर नथिंग बट द स्टोर्ड फॉर्म ऑफ व्हाट? द स्टोर्ड फॉर्म ऑफ ग्लूकोज. सो ये रसगुल्ला का भाड़ है. कम ऑन, हैविंग मेनी ऑफ द रसगुल्ला. सो इंसुलिन क्या करेगा? इंसुलिन इज गोइंग टू ब्रेक डाउन दिस पर्टिकुलर भाड़ एंड लेट ऑल द रसगुल्ला कम आउट. दिस रसगुल्ला आर नथिंग बट व्हाट? ग्लूकोज. सो ग्लूकोज एनरिच हो गया इनसाइड द बॉडी. बट दिस glucose should get inside the cell so that the cell gets the glucose and gets energy by breaking down what? Glucose. By breaking down what? Glucose. So C6H12O6 in presence of O2 is going to give rise to what? Carbon dioxide plus water. Along with that 686 kilocalorie of energy is going to come out, which corresponds to 38 ATP. Come on, we have done it already in respiration topic. But from the blood vessel glucose अंदर कैसे आएगा? Who is going to tell me? Class 11 biomolecule topic. There was a receptor. Yes. Class 11 biomolecule topic.
Active.
No. A receptor which can help in active transport of glucose. Come on, help me. What is the name? GLUT4.
GLUT4.
Very good. GLUT4. GLUT. GLUT4. Fine. So this GLUT4 receptors are present over here, which is going to allow the glucose entry. But ये तभी आएगा जब इंसुलिन इसको हेल्प करेगा. If insulin are going to come and bind with the cells, then only ये दरवाजा खुलेगा so that glucose enters inside. Again I repeat. Every cell is having insulin receptors. But blood vessels से सेल के अंदर तब जाएगा where it is only going to come inside the blood, inside the cells, if the insulin receptors are attached with the cells. If the insulin receptor, the blue one are insulin receptors, if they are attached with the cells, then only it is going to allow the GLUT4 open up and glucose is going to get inside. बोलो समझ में आया क्या? So ग्लूकोज अगर एंट्री ले लेगा सेल के अंदर, ग्लूकोज ब्लड वेसल्स में ज़्यादा रहेगा कम रहेगा? कम रहेगा. The glucose level is going to come down and that is a function of insulin. बोलो समझ में आ रहा है? अब आपका बॉडी बन गया इंसुलिन रेजिस्टेंट. क्या समझ में आएगा? इंसुलिन रेजिस्टेंस मतलब क्या है? इंसुलिन रेजिस्टेंट बॉडी कैसा होता है? इंसुलिन थोड़ा सा इन्फ्लामेटरी बॉडी होगा. क्या होगा? इन्फ्लामेटरी. इंसुलिन रेजिस्टेंट. That means ग्लूकोज उनके बॉडी में क्या रहेगा? ज़्यादा रहेगा कम रहेगा? Okay, we are not going to talk about that, but got the idea what is the function of insulin? Now normally before 1980 ये इंसुलिन किसके ऊपर डिपेंड किया जाता था? Slaughtered cattle and pig. Slaughtered cattle and pig. Is this clear? But after 1920 we have started making insulin by the help of bacteria, and this was done by Eli Lilly company, an American company. तो बॉडी में किस फॉर्म में रहता है? A, B and C. There are 3 polypeptide chain. 3 polypeptide chain. प्रत्याशा, what is the meaning of polypeptide? That means they are made up of what? Many amino acids. But the presence of C is going to make it inactive, called proinsulin. So what has to be done? Which two is going to make active one? A and B. A polypeptide chain and B polypeptide chain is going to make it active insulin. So we have to remove this C part. So A and B, this A and B polypeptide chain, the DNA which makes the DNA of the pancreas cells, the DNA of the pancreas cells which can make, this is mRNA, and which can make the insulin, which can make the insulin. Which part of the insulin? The A polypeptide chain, the A polypeptide chain जो बनाता है, that particular part, this is A polypeptide gene, clear? So this part of the DNA has been cut and introduced where? Is being introduced where? In that bacterial plasmid. In that bacterial plasmid. This is the gene of interest now. Making what? This is the gene which can make a polypeptide chain. And then we have put this particular, so we have made this rDNA. We have made this rDNA inside the bacteria, rDNA inside lab, and put it back inside the bacteria, allowed the bacteria to grow, and then put it in the bioreactors and found what? Only a polypeptide gene in a large scale. Same way, what we have done, और एक कौन सा है? B polypeptide chain, that particular DNA or gene which can make, that particular gene which can make the B polypeptide chain, this is B polypeptide chain. This DNA has been cut and introduced where? Inside the plasmid, and this is the particular B, the gene which can make B polypeptide chain, and we have made A separately and B separately. We have made A separately, this is the A chain, and say this is the B chain. And we have made a disulfide linkage and made what? Made humulin. Made what? Humulin. We have made humulin. Is this clear? So this is A1 and this is B1. And this humulin is now marketed in entire India. It is marketed. So out of 12 products, this is one of the product. Out of 12 products which is marketed in India, biological product for medicines, one of this product is humulin. One of this product is humulin. Just wait. Let's complete insulin. Humulin. Let us complete humulin. Yes. Yes. Vaccine safety. Yes. Humulin, ELISA, molecular diagnosis. Okay. Now, before gene therapy, we have got humulin. This is humulin. This is humulin. See. Okay. Genetically engineered insulin. All time is up. It's 9:33. Yes. Can we start this in the next class?
Yes, sir.
Huh? Just have a look about this topic, go line by line of this genetically engineered insulin. Next day we are going to start from here only. I'm going to again give you some of the information about humulin that we have talked about lastly. And then the next topic is going to be about humulin, gene therapy, and we have to complete this. Vaccine safety is there. Fine. Rosie the cow is very important. Fine. Rosie the cow. ऐसा एक मूवी का नाम है ना, दाग द फायर. So Rosie the cow. So this cow is going to be very important for us. Fine, we will talk about that. Chalo, that's it.